Hello everybody. Just like we said in class, since I'm out of town, we're going to handle the challenge questions slightly differently this week. I'm asking you to go ahead and give these a try first and submit your answers to me. And then you can watch this video to see the correct solutions done for you, demonstrated for you by me. So, like the challenge questions in class, you'll get full credit whether your answers are right or wrong. So please don't feel tempted to watch the video first and then write your answers just so you get them correct. I want to see your best try, and you'll get full credit for your best effort. So if you haven't done these already, please pause the video right now and give these challenge questions your best try. Submit them through Canvas so I can see how well you did on your own, and then definitely watch the videos to see me demonstrate those best answers. So here's the first challenge question. Really focuses on the central dogma. And this challenge question is all about determining the expression of a gene. So first we need to make that gene into messenger RNA, and then we take that messenger RNA and translate it into a protein. So the first thing to point out here is a typo. I know that I'm not looking at mRNA in this sequence, and take a moment to think about why that might be. The reason I know I'm not looking at mRNA is because I see thymines. So this should read that you are looking at the DNA for a gene. And A asks you to give the mRNA sequence and the amino acid sequence for the gene that this encodes. Well, the mRNA sequence is easy. So transcription as a word just means to copy in a different form. Medical transcriptionists take audio recordings by doctors and nurses and PAs and type them into written reports. Legal transcriptionists take the proceedings that are spoken in a courtroom and type them into written notes. In all of those cases, the language is unchanged. We're just changing the form from audio to typed, or the other way around. The reason why transcription is called transcription is because the same rules apply. The language of DNA is the language of nucleic acids, and when we transcribe DNA, we stay in the same language. We change to messenger RNA, to an RNA molecule, but RNA is still made of nucleic acids. So transcription transcribes, and it changes DNA into RNA. The only real primary difference between DNA and RNA in terms of sequence is that RNA has a little bit of an accent, and it pronounces its T's as U's. There are no thymines in RNA. Instead, thymine is replaced with uracil. So in order to transcribe the above DNA sequence into RNA, we simply rewrite the sequence exactly as we're given it, except we change all of the T's, the thymines, to uracil or U's. So this is the transcribed version of this DNA gene. My apologies for the handwriting. The stylus doesn't do fine print all that well. But the sequence is AUG, GCA, CAG, AGG, GAA, CUA, UAA, CUU, GAU. And that's all there is to transcription. Translation is changing languages, just like we mean the word in regular speaking. If we translate, we're going from English to Spanish, from Chinese to Portuguese. We're changing the language. And translation in molecular biology is the same exact thing. Here we move from the language of nucleic acids to the language of amino acids. And to do that, we need a few extra tools in our back pocket. First, we need to realize that each set of 3 nucleic acids in DNA or RNA represent a single amino acid. So we're going to go from 3-letter words to 1-letter words when we translate. 2, just like we need a dictionary to translate languages, an English to Spanish dictionary, a Spanish to Chinese dictionary, we need a dictionary to translate the language of nucleic acids to the language of amino acids. And that dictionary is called the genetic code. So in order to decode or to translate this mRNA sequence that we just deduced, we're going to need the genetic code. We're going to need the nucleic acid to amino acid dictionary. And I will provide you with a genetic code on the 3rd exam, and on the final. You do not need to memorize the genetic code. No one should have to memorize the genetic code, and so that will be provided for you. So here is the genetic code on the screen. I've recopied the the nucleic acid sequence of the messenger RNA at the top there, of what we just had deduced a moment ago. And so now it's time to decode or translate this sequence. So the way that we use this dictionary, this genetic code, is we start with the first letter here on the left, we use the second letter at the top to narrow down our search, and then the third letter corresponds to what we see on the side here. For example, if the first codon or triplet that we're trying to translate is AUG, we start with the A's here. The next letter is U or T, so now we're down to this box here. And then finally we look for the G as the third position right here. That means it's the last position here. So methionine is the first amino acid. And I should say that methionine is always the first amino acid of every protein for every single cell that lives on this earth. The AUG start codon starts all proteins. So this starts the translation. This establishes the reading frame. The next codon we see is GCA. So it's GCA. And so GCA is found right here, and it codes for alanine, ALA. You don't have to know what the single letter or triple letter codes of amino acids stand for. In other words, you don't have to know that this is alanine. You'll be required to learn that in biochemistry. The next codon is CAG. So CAG is right here. That's glutamine. And I'm going to go ahead and clean my table up a little bit so I can continue decoding. And then we have AGG. So AGG is right here. That's arginine. GAA. GAA is right here. That's glutamate, or glutamic acid. CUA. CUA is right here. That's leucine. UAA. UAA lives right here. That's the stop codon. And then we're done. Once we hit a stop codon, translation ends. So don't even concern yourself with these triplets or codons, because stop means stop. The translation is over. And so the amino acid sequence of the protein that this gene encodes is methionine, alanine, glutamine, arginine, glutamate, leucine, period. Done. That's it. So that's what we're looking for there in that question. So let's go ahead and attack the next set of questions by mutating this sequence and seeing what happens. I'm going to first go ahead and draw in the amino acid sequence that we've already determined, and then we can compare each of these individual point mutations to that original sequence. Now the first mutation that we're challenged with is a transversion to cytosine at nucleotide 11. So let's find nucleotide 11. It's right here. If this A is nucleotide 10, then this is nucleotide 11. And we are undergoing a transversion to cytosine. Now a transversion, if you remember, is when we switch nucleotide classes. So the purines are adenine and guanine, and the pyrimidines are cytosine and thymine. The reason why we're told cytosine in this case is because if we have a transversion of a guanine, we're switching from a purine to a pyrimidine. We're switching from guanine to either cytosine or thymine. So I had to tell you which of those two you are transverting to. So that's a transversion to cytosine at nucleotide 11. That changes that codon from AGG, which in this case was our arginine. It changes from an AGG to an ACG. An ACG is a threonine. ACG is a threonine. This is a missense mutation because we're changing from one amino acid, an arginine, to another amino acid, or threonine. So the phenotypic effect of this is that it is a missense mutation. The second mutation is a transition of nucleotide 12. So let's not make these bulk up, so we'll go back to here. And now it's a transition at nucleotide 12. Nucleotide 12 is this second G here. And we're not told what nucleotide we are mutating to because it's a transition. Transitions are when we remain in the same purine or pyrimidine class. Guanine is a purine. The other purine is adenine. So this mutation is essentially telling us that we are changing this guanine to an adenine. We are changing from AGG to AGA at the same exact codon. Well, AGG codes for arginine. We knew that already. And AGA also codes for arginine. So this mutation falls into the class of silent mutations. Silent mutations are where, although there is a point mutation at the DNA and RNA level, it doesn't change the amino acid that the codon codes for. And since life is all about the proteins that we make, and the proteins really govern life, if the amino acid sequence is not changed, if the protein is not changed, then we will not have any consequential effects of that mutation. We refer to it as silent. The third is a mutation at nucleotide 7, and it is also a transition. So first we have to find nucleotide 7. Well, this cytosine is 5, so let's go to 2 away from it. So it is this cytosine here that I'm underlining in green. And it's another transition, so we're staying in class. So this cytosine is going to change to the other pyrimidine or thymine at the DNA level, uracil at the RNA level. So that's this glutamine here, this third amino acid, has changed from CAG, which again is glutamine, to TAG, or UAG on the genetic code, and TAG is a stop codon. So this mutation actually turns this glutamine to a stop codon, and that prematurely truncates the entire chain of amino acids. Remember, stop means stop. So if we change this glutamine codon to a stop codon, we never translate the arginine, the glutamate, or the leucine that comes after. This particular type of mutation is referred to as a nonsense mutation. Nonsense mutations change codons that initially coded for amino acids. It changes them to stop codons, prematurely truncating the protein completely. Finally, the last example of a mutation that we have here is an indel. It's an insertion or a deletion. And in this case, the indel is the assertion of a gene, of a G, immediately following nucleotide number 10. So nucleotide 10 is this adenine right here, and this insertion is putting a new G in that place. Let me draw that again so it's a little bit more obvious. It's inserting a G in this area. Now this is going to result in what's called a frameshift mutation. It's going to reestablish a new frame of translation. This is certainly still the first codon. This is the second codon. This is the third. But now the fourth codon is AGG here because of the insertion. That's the set of 3 nucleotides. And now our frameshift. This is the next codon, GGA, and then this one, and then this one, and then this one. And you can see the entire frame has now shifted from the insertion all the way on down. We can see the effect of this frameshift mutation by retranslating the entire reading frame. We start with the ATG, so we have methionine once again. The next codon has not been altered, so after methionine is an alanine, that's the GCA. Following the alanine is still glutamine, that's the GLN amino acid from the CAG codon. And then we don't even change the arginine because our initial codon was AGG, but we inserted a G. The new codon in that boxed position is still AGG, and so we still have an arginine. So far, no change. And if there's no change to the amino acid sequence, so far no effect. The next codon after the frameshift is GGA. Now if we look at our amino acid to nucleic acid dictionary, we see that GGA codes for glycine. Well, that's new. We had glutamate before, and now we have glycine in there. That can't be good. And then next we have an ACT. ACT codes for threonine. Ooh, that's not good either. In biochemistry, you'll learn that threonine is very big and bulky and somewhat hydrophobic. So that's going to change things quite a bit. And after that ACT3, threonine, we're going to have an ATA. Well, ATA codes for isoleucine. That's new. And after the ATA, we're going to have ACT. ACT, oh man, another threonine. We have a much longer protein now, and it contains amino acids that we never had before. And then we have a TGA, or a UGA in the mRNA. And TGA's codes for stop. Thank goodness we're done with this mess. And so what we can see here is that frameshift mutations actually cause a shuffling or a randomizing, an absolute radical change of the amino acid sequence from the point of the insertion down. Frameshift mutations tend to be some of the most consequential and detrimental mutations that we can have. And it's because they scramble the entire amino acid sequence from the point of the insertion or deletion downstream into the gene from that going down. The reason for this absolute scrambling is in the name of the type of mutation itself. It shifts the reading frame of the codons. And by shifting that reading frame, we alter every single codon from the point of the insertion down. So the first challenge question here, just introducing you to not only the central dogma, but to the nature of mutations, a really, really nice way to bridge the two lectures from this week together, starting off by transcribing a DNA sequence, then translating that using the genetic code, and then using the terminology of mutations, such as transversion and transitions, to determine exactly what's going to happen at the RNA and DNA level, and then using the genetic code to translate those mutants, and using our other set of terms to characterize those mutants, such as missense, nonsense, silent, and frameshift.